Good morning, dear friends and students. I am Dr. Suchita Bharambe, Assistant Professor, Department of Microbiology, Government Institute of Science, Chhatrapati Sambhajinagar, welcomes you in today's session on AI and ICT in microbial analysis, which focuses on how technology is revolutionizing the study of microbes. In this session, we will learn how AI and ICT tools improve microbial identification, its role and role of bioinformatics in analyzing microbial DNA. It also emphasizes on comparison of traditional versus digital methods in microbiology. Analyze the real world application of AI in disease detection and evaluate the benefit and challenges using this technology in labs. Before we start, we should know about certain key vocabularies. Terminologies like artificial intelligence. Artificial intelligence refers to the computer system designed to imitate human thinking abilities such as learning, reasoning, pattern recognition, and decision making. In science and education, AI helps in analyze large datasets, identify trends, predict outcomes, and support accurate decision making. Another terminology we must know, that is machine learning, which is a process in which computing systems learns from the data and use algorithms to execute tasks without being implicitly programmed. The next terminology, that is ICT, information and communication technology, which includes digital technologies like computers, software, mobile devices, and internet network used to collect, store, process, and share the information. In teaching and research, ICT enables online learning platform, virtual laboratories, data sharing, and effective communications. According to the demand of today's session, along with the ICT and AI, one must know what is microbial genomics. Microbial genomics is the study of complete DNA of microorganisms such as bacteria, fungi, and viruses, which helps to understand their genetic makeup, metabolic pathways, virulence factors, antibiotic resistance, and evolutionary relationships. To merge these things, we should know what is bioinformatics. Bioinformatics use the computers to store, analyze, and interpret biological data, especially DNA sequences. The traditional versus digital microbiology, where old ways, old methods were taken over by the digital technologies for identification of microorganisms. The diagnosis is an important factor in the healthcare, and it is essential to identify microorganisms that causes infectious diseases. AI systems can improve disease management, drug development, antibiotic resistance prediction, and epidemiological monitoring in the field of microbial diagnosis. AI systems can quickly and accurately detect infections, including new and drug-resistant strains. It also enables early detection and antibiotic resistance and improve diagnostic techniques. Thus, the application of AI in bacterial diagnosis focuses on the speed, precision, and identification of pathogens, the ability to predict the resistance, In traditional microbiology, scientists relied mainly on light microscope, petri dishes, staining techniques, and manual observations to study the microorganisms, where the identification of microbes based on colony morphology, biochemical test, and visual interpretations, which require skilled personnel and considerable time. This makes these methods slow and labor intensive and limited to what could be directly observed by the human eyes only, making detection of subtle and early changes difficult. But with the integration of ICT and digital technologies, microbiology has transformed significantly. Digital imaging systems provide high resolution, visualization, automated sensors and instruments, enable continuous monitoring and cloud-based databases, store and share massive biological databases. The techniques such as automated microbial identification, genomic sequencing, and AI-based image analysis allows the rapid, precise, and reproducible results. This shift from traditional to digital microbiology matters. Digital microbiology reduces the human error, speed up the diagnosis, and improve accuracy in identifying pathogens. It enables the real-time data sharing across laboratories and countries, which is crucial during the disease outbreaks and pandemics. Overall, the digital tools enhances efficiency, support evidence-based decisions, and strengthen the global public health responses. Identification of microorganisms through the techniques as culture, molecular analysis and imaging starts with appropriate sample collection and run into several problems with the conventional procedures including sample handling, difficulty in culture identification, incorrect identification may be there. The microbial susceptibility testings are difficult. These traditional methods requires manpower and treatment for delayed time. So integration of AI in this traditional analysis may fasten up the identification processes. High resolution images of microbes are taken nowadays with digital microscopes or mobile apps. AI analyzes the shape and size, color pattern of microbes to classify them in the group. It helpful in the taxonomical analysis. One of the best example in the where the machine learning models could help, such as DNA sequencers, which analyzes the genomic sequence of bacteria and viruses, which is helpful for identification of these organisms using the global databases, helps to identify them as species level. Also, it could predict the tendency for the mutations in these organisms, the resistance to the specific drugs. It guides the clinicians in selecting the most effective treatment options. The AI-powered automation speed up the process such as sampling processing, data interpretation. This not only shorten the diagnostic turnaround time, but also allows healthcare professional to concentrate on the patient care. Nowadays, microscopes enhanced with AI have potential to help microbiologists in examination of microorganisms. In a study, microbiologists at Beth Israel Deacon's Medical Center have demonstrated that an automated AI enhanced microscope system highly adept at at identifying images of bacteria quickly and very accurately. To implement AI in microbial analysis, the one indispensable part is bioinformatics. Bioinformatics is an interdisciplinary field that combines biology, computer science, and statistics to understand and interpret microbial DNA. It allows the scientists to convert the complex biological information in meaningful or actionable knowledge. Modern DNA sequencing machines read the nucleotide bases A, T, G, C in microbial genome. These machines do not produce biological samples as output. Instead, they generate large volume of digital data representing in genetic code of microorganisms. The raw DNA sequence is processed using bioinformatics softwares and algorithms. Involves cleaning and organizing sequence data, computing sequence with reference genomes stored in digital database, helps in identifying mutations, gene variants, and resistance markers in the organism's genome. By comparing DNA sequence, scientists can track how pathogens spread across the region. By comparing DNA sequence from different patients or locations, scientists can identify whether infections are caused by the same strain. They can trace how the pathogens speed up across the region of communities, detect sources and transmission pathways during the outbreak. This is especially helpful for controlling the infectious diseases. Say example, in India, bioinformatics helps in identifying the drug resistance tuberculosis strains very quickly. AI-based uh, bioinformatic tools predict drug resistance within day by detecting specific genetic markers. This result of bioinformatics analyze and helps doctors to choose the most effective treatment as early as possible to reduce the treatment failure and drug misuse also. Thus, it saves lives and limit the spread of drug-resistant TB all over the region. This is one of the AI-based instrument nowadays available in market. The intelligent imaging and machine learning technology to read and interpret the presence of significant bacteria in culture plates. When the culture plates put into the specific area of the machine, it analyzes the colony characters, the morphology, and also it go for the biochemical testing for identification of microorganism in a very little time. The real world AI tools are also available in the markets. The AI powered microscopes combines the digital imaging with the machine learning algorithms to automatically analyze the microscopic samples. For example, tools like PathoSpots can scan the blood smear and detect malarial parasite by analyzing their shape, size, and staining patterns. They compare if we compare it with the manual microscopy, this system is faster and more consistent, could give the reproducible result and reduces the burden and resource limited settings in certain areas. Along with that, number of mobile apps are also available for the field diagnosis. The AI enhanced mobile applications such as Bactoscan allowed the field workers or the technicians to capture the images of sample using smartphones. They can upload them on the AI systems and receive the instant microbial identification or preliminary identification analysis of that microorganism. These are specifically valuable in the remote and rural areas where laboratory facilities are limited. They enable the rapid screening, early interventions, and better disease surveillance at grassroots level. Another tool like Cloud Lab is there. Cloud Lab based platforms like BioCloud India allows the researcher to perform the data analysis and simulations, online simulations to access by accessing the global genomics and microbiological database. It allows the collaboration with the scientists across the institutions and countries in real time instead of running every experimental locally. Researchers can save the time, cost, and infrastructure while importing the data, sharing responsibilities, and also it provide the reproducibility of the results. Why these tools matters actually? Together, smartphones, mobile apps, and cloud labs, they speed up the diagnosis, reduces the human error, enable the real-time data sharing, and strengthen the responses to infectious disease outbreaks. Thus, the AI tools transform the microbiology from manual, location-bound work into fast, accurate, and globally connected science. So, whatever we discuss up till now, I think you get a basic idea about how AI tools could be useful in microbial analysis. So you could answer these simple questions like what is microbial genomics and how does it supports disease control and in public health? Yes. How does bioinformatics transforms raw data into actionable medical insight during an outbreak? You could compare the traditional microbiology techniques with the digital methods in terms of speed, accuracy, and scalability. These are the answers to the questions. You could compare with your own answers. Though AI and ICT is useful, it have two sides, advantage and challenges also. It have benefits and certain challenges also. Though AI based analysis, it is faster, it gives the early outbreak detection, it allows the remote access via mobile tools, it reduces the lab workload, but it requires high equipment cost. The reliable internet availability is the mandatory part. Trained staffs are required to utilize these apps, and that data validation is the crucial to prevent misuse and error. But if one this these challenges handled very properly, it is a very useful technology. AI and ICT are transforming microbial analysis, making it faster, smarter, and more accessible, from identifying bacteria with smartphones to tracking disease outbreaks using DNA data technologies. It's saving lives, while challenges remain in the future of microbiology is digital connect and full of possibilities. Thank you.